Students, I'll discuss about 2021 Neet Biology Final Exam key. Right? In that, we'll see the first question from the botany. Which of the following statements is not correct? See, they're given 4 statements. Read the 4 statements thoroughly, and then you have to identify the which statement is not correct. So it means you have to identify all the correct statements. Now see, student, here the statement number 1 given: pyramid of numbers in a grassland ecosystem. In a grassland ecosystem is upright, right? And second option, pyramid of biomass in C is generally inverted. And third option, the pyramid of biomass in C is generally upright. The pyramid of energy is always upright. This is the fourth option. Among these 4 options, you need to identify the which option is not correct. Which option is not correct? It means incorrect option. See, student, before going into that, we'll see the different types of pyramids, different types of ecological pyramids. Now see, student, here, pyramid of numbers in a grassland ecosystem. In a grassland ecosystem, they are upright. See, this pyramid is upright pyramid, right? It means in a grassland ecosystem, when you take the numbers, see, in the trophic level 1, the number of individuals or more, right? And compared to trophic level 2, the number decreasing. When you are going from the T1 to the T4, the number decreases. The number decreases. The number of individuals decreases. That's why here we'll get the upright pyramid. So in a grassland ecosystem, the number of pyramid, it is upright. Now see the pyramid of biomass in aquatic ecosystem. Aquatic ecosystem, it means in oceans, sea, and then ponds, lake, whatever it may be, the aquatic body, where see the biomass, it means the weight of a particular trophic level. Now see, students, in T1, the biomass is less compared to the upper trophic levels, right? Where the biomass is more. Here the biomass is more biomass. When you go from the bottom to the top, it means from the primary or T1 to the T3, the biomass increases. The biomass increases, right? So in this pyramid, it is correct. This pyramid is inverted pyramid. Inverted pyramid. It means it is correct. In aquatic biomass, the pyramid is inverted, right? Now see the pyramid of energy in any type of ecosystem. The pyramid of energy in any type of ecosystem, that is upright. That is upright. Whatever the type of ecosystem, it may be grassland or aquatic, right? Wherever the pyramid is upright, it means see from the T1 to the T2, T3, T4, that is trophic level 1 to the trophic level 4, what happens? There is a 10% law. 10% law. It means from every trophic level, the energy flow will be 10%. It means from here, if it obtained the first trophic level 100%, from here only it passes 10%. Now this 10% is 100% for this T2. Now from here it passes again another 10%. From here again 10%. So from every trophic level, there is a energy flow that is just only 10% will be flowing. So it means what? The pyramid of energy in any ecosystem also upright, right? Now we have seen the different types of pyramids in different aquatic ecosystems. Now again we'll go to the options. Once again we'll check the options, then select incorrect option. Right. See student, once again read the options. The pyramid of numbers in a grassland ecosystem is upright. This option is correct option, right? And see, the pyramid of biomass in sea is generally inverted. See, C means the aquatic ecosystem, that is inverted, right? That is also correct. And see, the pyramid of biomass in sea is generally upright. See here, this option is not correct. Pyramid of biomass in sea is not upright, that is inverted. So this is the third option is not correct. And see the last option, that is fourth option, pyramid of energy is always upright. This is also correct. So for this question, which option is not correct? The third option is not correct. So the answer is option 3, right? Okay. Now let we discuss about second question. Second question, just will move the second question. See student here, question number 2. Right. See, which of the following plants is monoecious? Monoecious. See, first thing we should know about monoecious and dioecious. See, monoecious means if male and female flowers or male and female sex organs present in the same plant, then you can call it as monoecious plant. If male flower is one plant and female flower is in another plant, then you can say that is dioecious plant, right? Now see the options. We have to identify the monoecious plant where both male and female sex organs present on the same plant. Now see the options. They have given 4 options. The one is Cycas circinalis, Carica papaya, Cara or Chara, Markantia polymorpha. Markantia polymorpha. Right? Among these 4, you need to identify which is monoecious plant. Right? Okay. We'll see there before going into the find out the answer. Just we'll go and then see their different types of plants. Now see students here. See this is Cycas circinalis. Cycas circinalis, right? Belongs to the gymnosperms. Gymnosperms. Now see student here, separately there are 2 different plants. One is the male plant, right? The male plant bears only male reproductive structures and the female plant bears only the female reproductive structures. See student, as I first told that if 2 different plants are there for the male and female, then you can say that they are dioecious plants. They are dioecious. They are not monoecious. It means Cycas circinalis is not monoecious. It's not monoecious, right? And see another plant, Carica papaya. Carica papaya. Now again in the Carica papaya also there are 2 different plants in this species. In this species, 2 different plants. There are separate plants for the male and separate plants for the female. Now see here, in the Carica papaya, the female plant. You can easily find the female plants of Carica papaya where fruits are present. Fruits are present in female plant. In male plant, there is no fruits. No fruits in the male plant, right? So, and then again, Carica papaya, also dioecious plant. Also dioecious, right? It is not monoecious. And then you see that another option what they are given, Markantia polymorpha, or we'll see the Chara or Cara. Now see student, in a Cara, it actually belongs to the algae. It is algae. In this algae Chara, in the same branch, in the same plant, you can find the woogonium. The woogonium, this is female sex organ, and then antheridium, that is male sex organ. See, in this plant, in this Chara, both both male and female sex organs present on the same plant. So then this plant is monoecious plant. This is monoecious, correct? Okay. And see that again the Markantia, this Markantia belongs to the bryophytes. Now see, these are actually gametophytes. In bryophyta, the dominant body, that is gametophyte. Now see student, here there are two different types of gametophytes. There is a male gametophyte and female gametophyte. See, male plant bears only the male reproductive structures. They are antheridia. Actually antheria 4, here is antheridias, right? And then the female plant, the female gametophyte bears archegonia. Like archegonia produces ova, and then antheridium produces sperm, like male gametes. So it means this Markantia polymorpha also not a monoecious plant. This is also dioecious. See that now we have seen 4 different types of options, right? Carica circinalis, it's not a monoecious. Carica papaya, not a monoecious, right? And also Markantia polymorpha, not a monoecious. Carica circinalis is not monoecious, right? Carica papaya, not a monoecious. Chara, monoecious. Markantia polymorpha, not monoecious. So which of the following plants is monoecious? For that, the correct option is 3. Third option, right? Okay. Now see students, we'll see then third question. See that the first stable product, question number 3, the first stable product of CO2 fixation in sorghum is, right? In sorghum. See, first thing we need to find out the sorghum is a what type of plant. Either this is C3 plant or C4 plant, right? See, the sorghum is C4 plant. Sorghum is C4. It means this the C4, C3 given based on the formation of first stable product in the cycle, right? If it has 3 carbons, then we can say those plants comes under C3. If first stable product in the cycle is contains 4 carbons, then those plants come under C4, right? Now see here the options they have given: phosphoglyceric acid, pyruvic acid, oxaloacetic acid, succinic acid. See student, actually before going into that, we'll see briefly the cycle. C4 cycle, as I told, sorghum is C4. Now see student, in case of C3 plants, here the first product which is formed at this point, right, that is, it has in C3 cycle 3 carbons, that's PGA. But in C4 cycle, this is the first stable product, oxaloacetic acid. Oxaloacetic acid, it has 4 carbons in its structure. How many carbons? 4 carbons in its structure. So it means here, if you see, this is the entire cycle of the C4 occurs in 2 different places of the leaf. It means 2 different types of cells in the leaf. Now see the options once again. The phosphoglyceric acid, the PGA. This you can find in the C3 cycle. And pyruvic acid, you can find the end product of the glycolysis. End product of glycolysis. Product of glycolysis. See, one glucose molecule when it is getting breakdown gives 2 pyruvic acids or pyruvates. And see the oxaloacetic acid. And see student, this oxaloacetic acid is the initial molecule in the Krebs cycle, as well as this is the first stable product in the C4 cycle. And see student, succinic acid, you can find this as intermediate compound in the Krebs cycle. In the Krebs cycle. See here, among these 4 options, as you know, the sorghum is C4 plant. C4 plant. Now see, in the C4 plant, the first stable product, that is oxaloacetic acid. OAA. OA. Again, the correct option is third here, according to the options. Right? Okay. Then we'll see the fourth question. Right. See, student, here, the didelphous stamens. The question is, the didelphous stamens are found in didelphous. See, student, this is regarding about stamens. In how many bundles they are there in the flower? How many bundles? It means if they are in single bundle, they are called monodelphous. Single, the only a big single bundle will be there. If it is in two bundles, right, didelphous. If in three bundles, tridelphous. Or if it is more, many, then we can say that polydelphous. Polydelphous. Now see that options given: China rose and citrus. Option number 1, and then 2, China rose, and 3, citrus, and 4, P. That is Pisum sativum. Now see here these conditions of the stamens in given flowers. Now see, student, in a China rose, Hibiscus rosa sinensis, where these stamens are and existed in single bundle. Single bundle. It means in China rose, it is monodelphous. China rose, monodelphous. All stamens or existed in the form of single bundle. Now see the citrus. In a citrus, they are present in the form of many bundles. See, these are all many different types of bundles. In citrus, the stamens are polydelphous. Polydelphous. You see, students, in pea plant, Pisum sativum, right? Total actually 10 stamens are there, and 9 in one bundle and 1 in separate bundle, like 1 plus 9, or you can also write 9 plus 1. 9 plus 1, it is in two bundles, right? In one bundle, 9 stamens. In another bundle, only single stamen is present. It means in the pea, the stamens are existed in the form of two bundles. So didelphous condition we can see in the pea plant. Now see, student, will go to the options. See here. So as we have discussed, in China rose and citrus, China rose monodelphous and citrus polydelphous. Now see the again second option, China rose mono and citrus poly. And then pea plant, see 9 plus 1, 9 plus 1. So here didelphous condition, right? So the correct option for this, the fourth option, right? Fourth one is the correct. Didelphous stamens are found in pea, not in China rose and citrus. In China rose and citrus are wrong. Then see the fifth question, question number 5. See, DNA strands on gel stained with ethidium bromide staining dye, when viewed under the UV radiation appears as, see the options given: bright blue bands, second option yellow bands, and bright orange bands, and dark red bands. Students, here actually when the ethidium bromide is stained with the DNA, it appears bright orange bands. Now see the here picture. You see here these bands which are visible here, the bright orange bands. This is DNA band, right? So these DNA bands are stained with the ethidium bromide, right? All are DNA bands. As you know that when we run the DNA on the gel, lower molecular weight fragments will move far away, right? And the higher molecular weight DNA fragments will move very slowly near to the gel, near to the well, right? See here, all these are appear orange bands, right? Okay. When stained with the ethidium bromide, ETBR. See, student, here again, once again, options. Bright blue bands, not the correct, and yellow bands, not the correct, and bright orange bands. This third option is the correct option for this question. And again, fourth is not the correct, not dark red bands. So option number 3 is the correct, right? Okay. Now let me see, students, another question. Question number 6, right? See here, plants follow different pathways in response to environment or phases of life to form different kinds of structures. This ability is called, right? Now see the options given: the maturity, elasticity, flexibility, plasticity, right? Now before coming into the options once again, we'll see a little explanation regarding this question. Now see, student, here, the ability of an organism to change its phenotype in response to different environments, termed as plasticity, is a particularly important characteristic to enable sessile plants. Sessile means not able to move, to adapt to rapid changes in their surroundings, right? Plasticity is a quantitative trait that can provide a fitness advantage and mitigate negative effects due to the environmental perturbations, yet its genetic basis is not fully understood. Now, which means what? If any response, in an environment, based on the response, plants are going to adapt. So that condition is termed as plasticity. Now these students will go to the options once again. Right, see. Plants follow different pathways in response to environment or phases of life to form different kinds of structures. This ability is called not maturity, not elasticity, not flexibility, that is plasticity. Option number 4, the correct option, right? Let me see, student, one more question, that is question number 7. See here, the match list 1 with the list 2. Now see the list 1, protoplast fusion, plant tissue culture, meristem culture, micropropagation. This 4 A, B, C, D, and list 2, totipotency, pomato, somaclans, and virus-free plants. I need to match exactly list 1 with the list 2. Now, before going to match, we'll see the different types of terms and their explanation. Now, see, student, here in a tissue culture, in generally the totipotency. What is actually totipotency? And that is the ability of an organism or a tissue to regenerate from a single cell, was recognized from observations of plant regeneration from injured tissues. Simply we can say that, and this ability is there only for the plant cell. It means the generation of entire new plant from a single cell, right? That is called, that ability is called totipotency. Now see, student, recognition of this biological principle credited to the German plant physiologist Haberland, who ruled in 1902 that each plant cell had the genetic potential to develop into a complete organism, a complete plant. Now see here, student, the method, the basic steps in the plant tissue culture technology. It means they are taking a few cells, a small tissue from the leaf, and they cultured in the plant tissue culture laboratory, and they developed a callus, and the callus separated and single cell culture, and finally they have developed an entire new plant, right? And once it is adapted, it can be transferred into the field. So it means with the help of the ability totipotency in a big plant tissue culture lab, you can develop a new plant. Right, now see the another explanation, that is protoplast fusion. Protoplast fusion. Now see student, in this protoplast fusion, what actually scientists did? See, they took Solanum tuberosum, that is potato plant. They took the cell from the potato plant, and in the same way Lycopers esculentum, tomato, from the tomato plant, they took the cell. Like, see, that means protoplast. What is protoplast? The plant cell without the cell wall is called protoplast, right? After taking the two protoplasts and they added chemical PEG, like polyethylene glycol. Now this PEG can help to fuse these two protoplasts. The protoplast from potato plant and the protoplast from the tomato plant. Once they fused, now see student, there is a formation of again hybrid callus and heterokaryon with two nuclei. The cell with two different nuclei. The one nuclei from the potato and the nuclei from the tomato. Now then, once it has grown properly, you see the growth of hybrid colonies and hybrid plants, like that is called pomato. It's called pomato. You see here the pomato plant, it has potato and also tomato, both will be there. So with the help of protoplast fusion, they developed a new hybrid, that is pomato, right? Okay. Now see the one more explanation. See the micropropagation. See what is actually micropropagation. Now see student, here is the clonal propagation of plants in closed vessels under aseptic conditions, sterile conditions. Inside the vessels, the plants are grown on culture media that contain nutrients and growth regulators and are described as in vitro, which means in glass, in test tube or beaker or flask or any other like bottles. See here, while they are doing this culture, the micropropagation, they can observe little variations. See, they are called somaclonal. Somaclonal variation is defined as genetic variation observed among progeny of plants regenerated from somatic cells cultured in vitro in the laboratory inside the test tube or in the bottles. So it means while doing the micropropagation, there are formation of little variations. They are called somaclonal variations. See students, another explanation. When if you want to produce a virus-free plant from the infected plant, it means a virus-infected plant, we can go for the meristem culture. Now see, when virus elimination is the objective, right? To obtain disease-free plants, we can take shoot tips up to the 10 millimeter are used for rapid clonal propagation. It means shoot tip which has the meristem cells. It has meristem cells. So meristem cells are rapidly dividing cells. So there, those cells are virus-free. If we take the meristem cells, the shoot tip cells, and if we cultured in a tissue culture laboratory, then you will get a plant which has which do not have any virus. So virus-free plants. To produce virus-free plants from the infected plant, right? Then you can go for the meristem culture. Right? We have seen all the different types of explanations for 4. And once again, you'll go to the question. Now see student, here we have to match exactly for right. Now see protoplast fusion. See, with the help of protoplast fusion, they develop pomato, right? Plant tissue culture. See, plant tissue culture, totipotency. With that help of totipotency, we can go to the plant tissue culture laboratory, then develop entire plant. See, meristem culture, that is virus-free plants, the micropropagation, that is somaclonal variations. It means here you see that A2 and B1 C 4, and D is 3. D is 3, right? See here, students, options are not typed properly. Now see inside here, A2, and A2, B, B1, and C4, and D3, right? D3, micropropagation, somacronal variation. Now the option number 4, correct? Okay. See another question, the one more question, right? See here, in the equation GPP minus R represents equal to NPP. In this question, what R represents? The options: respiration losses, radiant energy, retardation factor, environment factor, right? Before going into the options, once again, you'll see the explanation. See student here, NPP, net primary productivity. NPP is net primary productivity. What is that? Amount of stored chemical energy the plant has to use for the growth. This is directly proportional to the biomass. So whatever the energy which is used to increase the biomass, that is NPP. And what is GPP? Gross primary productivity. The amount of stored chemical energy the plant earns through the photosynthesis. The total amount of energy which was earned by the plant through the photosynthesis is called gross primary productivity. And NPP used to increase the biomass. Now what is actually R? R is respiration loss. It means, see, amount of energy loss through the respiration, that is in the form of heat loss as CO2, etc. Right? See, R is respiration loss. It means whatever the loss which was there through the respiration, right, in the form of heat or in the form of any other, then remaining energy will be used to increase the biomass. It means when GPP minus R equal to NPP, or you can write NPP equal to GPP minus R. GPP minus R, right? Okay. Now see here, once again, options. See. Right. See here, R represents respiration losses, not radiant energy, not retardation factor, not environmental factor. For this question, the answer is respiration loss. Now see student, another question, the one more question, that is question number 9. Question number 9, very easy. The production of gametes by the parents, formation of zygotes, the F1 and F2 plants can be understood from a diagram called, see, there is a net square, bullet square, punch square, and Punnett square. As you know that R.C. Punnett, who invented the board, that's Punnett square board or Punnett checker board, and that can be used to determine, right, all the possible phenotypes. Now see here, this Reginald Punnett is the scientist, was among the first English genetist. Punnett devised the Punnett square to depict the number and variety of genetic combinations. Now see here, simply this board is called Punnett square board, and this can be used to know all the genetic combinations in any type of cross, monohybrid or dihybrid cross. So the correct option for this question, that is Punnett square, right?